All right, welcome to Documenting Botanical Diversity, Part 1. So first of all, I just want to talk about herbarium specimens. I'm curator of ferns at the university and Jepson Herbaria. And these specimens kind of up until very recently represent basically the entire foundation of floristic and taxonomic knowledge. So reading a flora, reading something like the Jepson Manual or the eFlora, all of the information that we know about phenology. So when things are in bloom, when they're in fruit, distribution, knowing where things occur on the landscape is usually tied to a specimen collected in a very specific place. They're the basis for taxonomic study. Having a collection like the herbarium where you can just open a cabinet and all these things are organized by taxon and by geographic location, and you can spread them all out on a table and compare them side by side. Herbaria are really designed for being able to do that sort of work. Understanding the morphology, being able to take precise measurements, being able to understand three-dimensional figures, sometimes rehydrating things. This is a beautiful specimen by one of my favorite collectors, Lowell Ahart, who collected this Castanea and dissected the flowers in such a way that they could be mounted and all the bracts around the flower could be measured. And so this is a great way to prepare a specimen for a particular morphological or taxonomic study. And so there's a lot of thought that can go into preparing a specimen for these specific purposes. And ecological studies, there's a lot of ecologists out there who may not make voucher specimens, who probably ought to be making voucher specimens. And the reason for that is people do a study, in the 1990s, and our taxonomic concept of a certain plant may be very broad at that time. And then people, later on might discover that, well, in fact, these are different species that look really similar, but they have different chemistry and they share different pollinators. And there's a lot that we can learn. And then you go back to this ecological study and you say, well, which species were they actually looking at? So that is part of the reason that no matter what kind of research you're doing, having an actual specimen voucher is what makes that study repeatable and makes your results interpretable in the future when our understanding of biodiversity may have changed or is informed by new lines of evidence. And then another really big focus of herbarium specimens is looking at systematics, looking at relationships. So, the structure of DNA was discovered something like in the 1950s, and there's herbarium specimens that had been around for hundreds of years. So I personally was able to sequence DNA from specimens collected in the 1800s that those collectors, Cyrus Pringle would have had no idea of the ways that these specimens could be used in the future. And you can't get DNA from a photo. So being able to use specimens in new ways. Another thing people have been able to do is analyze pollen and look at atmospheric carbon. So there's just incredible things that you can learn from these specimens, and analyzing the soil that's left in the roots. So there's just a million different ways that you can learn about the world through specimens, and there's tons of ways that we haven't even thought of yet. So in the next 50 years, to try to imagine what people will be learning from herbarium specimens is kind of mind-blowing. So in addition to being the foundation of so much knowledge, herbarium specimens are really just beautiful objects. So these are collections made by Emily Dickinson that she just had a really a nice eye. This is a collection at Harvard that is digitized, but it's just beautiful to kind of see these compositions put together. And of course, we discourage mixed collections on a sheet just because it makes filing specimens challenging. But for her own personal use, there's just something about poets often make really nice specimens. And then looking through specimens too is just like an amazing way to feel connected to history. You just feel connected to people through space and time. And just imagining being on this trip to be a fly on the wall on Christmas Day, shortly after World War II in the far southern end of the Baja California Peninsula in Mexico with Annetta Carter, Annie Alexander, and Luis Kellogg just climbing through this canyon and collecting ferns. It's just like amazing to imagine being in that place. And there's Annie Alexander with her rifle slung over her shoulder, now immortalized with Lou up in the Campanile. And then the other thing about herbarium specimens is they can be a witness to global change and extinction. And so we think about extinction and biodiversity crisis as being, a lot of man-made changes that are happening, but in some cases, natural disasters can also wipe out species. So I'm just going to read this annotation label from Mike Price. When I published Diplasium pinatubicum in 1990, there was absolutely no inkling Mount Pinatubo, dormant for millennia, would violently erupt the next year, 1991, with devastating consequences, presumably exterminating this forest-dwelling fern. So this is an example. The best place to find a new species, a species that's new to science, is in a museum collection. So there's a lot of specimens. This was collected in the early 1900s, and it was only described in 1990 and then went extinct in 1991. So it's just kind of amazing to look at this specimen. And we have specimens of this species in the University Herbarium in Berkeley, and to just look at the specimen and know that this is the only record we have that this species ever existed on Earth. So, it's like, how do you put a value on something like that? So some of these specimens are just totally priceless and just fundamental to our understanding of diversity on Earth. And there's also something really personal about collecting specimens and revisiting them later. So this was kind of a fun thing that, I had just finished my undergraduate degree at Michigan, was traveling in the Venezuelan Guyana. And I just remember so clearly this beautiful day of taking my shoes off and wandering up this stream, at the base of this waterfall and collecting all these ferns this is Bruce Holst and Eurybia vivas. And, it's my notebook with my sketches of that river in that day in April 2006. So I was collecting and was an associate collector on this. And so you can see the date in my notebook and the location on the label, and an associate collector of this, what turned out to be the holotype of a new species that was described by Alan Smith, who is now my closest colleague and also curator of ferns at UC Berkeley. So it's cool to just kind of get this sense of looking back on your life, and the specimens you collect, you just never know what they're going to be or what you might discover or how they may be used in research. So it can be really fun and rewarding to just to collect specimens and then see them used by others. So now specimens are totally critical and have been foundation of our knowledge. And recently, with tools like iNaturalist, I think there's an incredible complementary source of data that works in tandem with specimens. So perhaps many of you have discovered iNaturalist already, or have it on your phone or have it on your computer. And if you don't have it yet, there'll be time today definitely to download it and try it. So it's basically a social network for sharing biodiversity data. So when you make an observation on iNaturalist with your phone, it records some key information. So that includes who you are, the observer, what you saw, and that can be what you think you saw, or it can be what the artificial intelligence suggests you may have seen, or what other people think you saw, the date when you saw it, and the time, some form of evidence of what you saw. So sometimes it's nice to go out in the forest and record a bird song, or you hear an insect or something, or you can take photos, up to 20 photos at a time you can include. and then also where you saw it. So it will record usually with depending on where you are, with a precisional accuracy of about 3 meters. So whatever the GPS on your cell phone is capable of, it will record that location. And iNaturalist is for all taxa. So it's basically organized in this giant hierarchy that includes plants, animals, fungi, all kinds of things. So that's one thing to keep in mind when you're out in the forest and you say, well, I don't know what that is. That's really cool. And you take a photo. If you know it's a mushroom, just indicating that it's a mushroom will help that help other people find it and identify it correctly much sooner than if you just take a photo and upload it and say unknown. So whatever taxonomic rank you feel comfortable identifying something, that will help other people find it and maybe identify it for you. And it's just really cool too to have all these data both taxonomically organized and geographically organized. So much like opening up the cabinets in the herbarium and seeing things kind of organized in these two ways, it's nice because it allows people who are maybe specialists in the flora of a certain region to be able to look at images or look at observations from that area, or someone who is a major taxonomic specialist. So, you know, there's a guy in the Philippines who's just an expert on bees all over the world. And so he just goes on and he can identify bees in California and all these other places. So it really helps kind of organize information in a way that people can find it. And there's been kind of an exponential growth to the use. This figure is already a bit outdated, but now there are over 150 million verifiable observations worldwide. So it's kind of astonishing to see how much data there is and the pace at which we are gathering more data. And so some of you may also have used Seek. So Seek is basically the standalone algorithm used by iNaturalist. And so it's interesting. I've tried using Seek and try using iNaturalist at the same time. And it seems like the model in iNaturalist might perform slightly better a lot of the time. And I don't know if it's just more informed by geographic information or what, but it's nice to try both. I really like to use Seek if I'm in a garden or walking around and there's planted things. I say, well, I know what that flower is, but I can't think of the name. And sometimes that's a really useful tool, especially for cultivated plants. And so I should also say iNaturalist was developed by a student at UC Berkeley in the School of Information. And it was sort of fostered by both National Geographic and the California Academy of Science. And now it is an independent nonprofit. And so when you make observations, so Seek, another reason to use Seek is if you're just casually looking at plants, especially cultivated plants, you don't want to necessarily share that data with the world or upload it or share any personal information. Say you're doing it with a kid who doesn't really have an online presence, that's a great tool. But if you're using iNaturalist and you are up for sharing your data, which is, I think, a really wonderful thing to do, you'll see this designation of a research grade observation. So basically what that means is to be verifiable, the observation will have a photo or sound, a date, and a georeference location. So, and then to become research grade, once it has those things, more than 2/3 of identifications have to agree. So if you say, well, I know this is Allium crispum, and someone else comes along and says, yep, that's right, that's becomes a research grade observation. So if somebody disagrees, then you have to have more people agreeing to reach that 2/3, more than 2/3 threshold. So it is a cool system. It's a lot like Wikipedia, where people had a lot of doubts about the veracity of the things that were being shared in Wikipedia, and as it's accumulated data and users, and it's become, this amazing resource. So it is cool. I really believe in crowdsourcing of data. I think it's a nice I think it's a nice approach, but obviously has some problems, and people do make mistakes. But the more people are able to see these sort of things, the higher quality the data become. So another thing to consider making observations is geoprivacy. So there are two categories basically. So well, of geoprivacy. So you can use 3 different designations. It can be open, which is the default. It can be obscured, where you say, well, you know, I'm on my friend's yard and you know, my friend's wetland and found this cool orchid and I don't really want people to come see it, or this is a rare bird, I don't want people to come and disturb its nest or something. You could obscure that location and it will give you about a 0.2 by 0.2 degree cell, or 500 kilometers squared. So it is not precise enough for someone to be able to find that organism, but it is still sort of broadly useful in understanding distribution of species. So it's kind of a nice middle ground. So you can obscure something yourself, or you can just make it private altogether and there is no location of any kind. And so that is not eligible to become a research grade observation. And then, so this is the geoprivacy that you can control yourself when you are making an observation. And then there's also taxon geoprivacy. So sensitive species will be obscured, and that is different depending on the species and depending on the geographic area where that's found. So something like Torrey pine, that is an endangered, very rare plant, but it is not, the location is not really secret. People know where to find Torrey pines and they're not really subject to poaching. People are not going on iNaturalist so they can find locations of those trees and, you know, try to dig them up and take them home. So some species, even if they're not altogether rare, but are subject to poaching, they may be obscured. And so this is done by curators, and it's a taxon by taxon, location by location sort of decision. So there's not one broad arching criterion that controls everything. It's kind of nuanced. And that's one of the cool things about iNaturalist, is that so many people can participate. So again, that's just the latitude and longitude with the obscured point is whatever the point is, it's just going to appear in a random place within that grid cell. So again, talking about the sort of decentralized system. So the taxonomic framework has been moving more closely towards adopting Plants for the World Online, which is the Q taxonomy. But there are some exceptions. In California, tends to follow the Jepson manual, and that's in large part because there's a lot of curators here, especially for taxa that are endemic to California. Another thing that's really wonderful too is if a new species is published, say a new species comes out in Madronio, we don't have to sit around and wait for somebody at Q to decide that it's a new species and add it to their database. You can just make a taxon tag on iNaturalist, notify a curator, hey, this new species was described, here's a link to the paper, and that species will appear in the iNaturalist database, you know, sometimes overnight or within hours. So it is really cool that it's a very nimble system. And the way that the discussions are on the taxonomic flags is there's a lot of transparency there, so people can kind of go back and see how decisions were made. And so yeah, it's a wonderful set of data that really complements natural history collections. And I talked a bit about herbarium specimens already, but you know, the things that you can't get from an iNaturalist observation are DNA sequences, and then almost never, you know, I work on this group of ferns that have these really intricate hairs and, you know, details of the veination and the glands. And it's like, you really need to get some things underneath a microscope to be able to confidently identify them. So these are the sort of things that are not always easy to work with on iNaturalist. So, you know, lichens can be tricky. Some things work better than others, but for a lot of vascular plants, an iNaturalist photo is often good enough to identify something to species. So another advantage, I'm just going to talk about some of the advantages, is the amount of data that is just increasing exponentially. So in the example of oak, for every herbarium specimen of an oak, there are 26 observations on iNaturalist. So you can start to get a finer scale understanding of plant distribution. But this does not come without bias. If you look at where both herbarium specimens are made and where iNaturalist observations are made, they're definitely concentrated around human habitation. So wherever a university or an herbarium is, you tend to have a higher concentration of herbarium specimens, and iNaturalist data are probably even more biased, with a huge proportion of urban observations. Another advantage of just having photos is that you're capturing all kinds of associated data without even having to try. So especially if you get a nice wide angle shot, you're going to get a lot of information about the habitat, some associated species. You may capture pollinators and herbivores. Sometimes you take a photo and you don't realize it. You get home and you see there's a beautiful pollinator visiting. And then also things like pathogens. So being able to kind of capture all this associated data that is just intrinsic to the photos. You can collect and share data in real time. So this is just a really wonderful way to conduct a BioBlitz, for example, where you may have a huge citizen science effort. People are all collecting at once and can kind of work together. And people like to gamify it or have a contest of who can see the most species in a short time or something. And then just as a tool for citizen science, John Redman has just done an amazing job with the San Diego County Plant Atlas. And this originally started as a volunteer effort with people collecting specimens. And then from that kind of grew into, or he transitioned into an iNaturalist effort. But part of that was dividing San Diego County into all these grid cells and seeing the places that had no specimens or no observations, and encouraging people to visit those places. So, and then John Redman has done over 600,000 identifications on iNaturalist. So because of his efforts and the efforts of all these volunteers associated with the museum, there's a kind of a remarkable fine scale distribution level understanding of the flora of San Diego County, which is really cool and is sort of been facilitated by iNaturalist. So that's one of the links that we shared before was there's a beautiful guide there of how to take photos for iNaturalist with a taxon specific recommendations. So for example, taking a photo of Asteraceae, you want to shoot the involucre, get the filaries at a side shot of the head, basically. And another thing that I really appreciate is that the data are fair, so findable, accessible, interoperable, and reusable. Another really nice thing is that you can really easily query the data, so you can look for the attributes that interest you and export them as a spreadsheet. So that's a really nice tool. And I'll just mention, if you are doing this through iNaturalist, usually aim for no more than about 1,000 records. So sometimes if you're looking in a small geographic area or for a rarer plant or your own observations from this 1 week period, doing it directly from iNaturalist is great. If you're doing more than 1,000 records, and that could be basically any information, you can go directly to GBIF, which is the Global Biodiversity Information Facility. And about once a week or so, all the iNaturalist research grade observations end up on GBIF. So if you're doing research with huge numbers of observations, you can really get a lot of data by going through GBIF. So one of the things, because they're research grade observations, you won't get the non-research grade observations where only one person put a name on something and hasn't reached that 2/3 agreement identification threshold. So, but just the way the data are structured, make them really easy to export as spreadsheets from either GBIF or directly through iNaturalist. We'll get into that a little bit later on. So another cool thing is just having this decentralized open source system for identification. So having an opportunity for essentially public open dialogue of annotations where people can see something and say, oh, maybe you want to revisit this identification. I'm noticing, something about it's cool to, people are able to sort of interact. And that New York Times article that I sent around about iNaturalist being the nicest place on the internet, I kind of agree with that. I described it as my favorite way to interact with strangers on the internet, is because people really do kind of have this shared goal of trying to understand biodiversity, and they're all using the same evidence. They all have the same facts of these images and are just using whatever resources are available to try to understand what that thing is and get a correct identification. So that's just sort of a nice thing. And then, unlike an herbarium specimen where the annotations may be in the folder and there's a lot of additional effort to either re-image the specimens or update those annotations, it's all kind of happens in real time and is really easily shared. And this also kind of extends to being able to communicate directly with other users. So in this case, somebody sees this cool aphyllon and says, well, hey, is it potentially on a gallium? So some of the diversity in aphyllon is very host specific, and identifying species depends on what host they're using. So it's cool. You can contact people and say, hey, was this other plant growing around, and have those sorts of conversations that people are often really enthused to take part in. And as more and more data are available on iNaturalist, there's a lot of global experts in various taxonomic groups who are using it. So Aaron, if you take a photo or some nice photos of an astragalus, Aaron Liston is probably going to come along at some point or another and help you put a name on it. Barbara Paris, who's curator of ferns at Kew, is retired now and has done over 100,000 identifications of ferns. Matt Williams is really great with Boraginaceae and helps to put names on a lot of those things and plants of the Channel Islands. So there are more and more experts who are using this resource. And another advantage over herbarium specimens is being able to non-destructively sample or document things. So this is a new species that was recently described. And on the right there, you can see this is the total global distribution of this species. And so you wouldn't want to go to all these places, and some of these places already are represented by herbarium collections, and make a new specimen to document these locations. So, with really rare plants that you don't want to over collect, it's really great to just have the photo and be able to monitor, so you could go to these places once a year and see how the populations are changing. Things like that. And it's interesting talking to students, and the way that people think about artificial intelligence is, this feeling that, okay, it's the robot that is learning to identify plants. It's the robot that's telling you what this thing is, and we can't trust the robot. This is a really interesting feature. So on this Calanthus over here, it says suggestions based on the observations and identifications provided by the iNaturalist community, including Ryan Odell, John Redman, Jim Moorfield, and many others. So those are all names of people I know. So this is not just a robot and a bunch of strangers. These are our friends and colleagues who are really putting in the effort to curate these data by identifying plants. And so it's easy to think about artificial intelligence as just being, this machine, but it's actually the people who are training the machine are experts who are really putting in a lot of time and effort and sharing their expertise by looking at photos. So I think it's helpful to think about that, and also to appreciate and be grateful for all the work that that folks are investing to make this platform really useful for everyone and to help expand this knowledge. And it's also a way that you can contribute by, the effort that you put in trying to identify observations, other people's observations on iNaturalist, or taking your own really excellent photos, as you are contributing to this shared knowledge, which is really nice. So, sometimes instead of waking up and doom scrolling in the morning, you could just wake up and over coffee, you could just, identify plants for 20 minutes while your coffee is brewing. And it's just kind of a nice way that anyone can contribute. And then this is another thing that's really cool. It was just announced, I think this week, is that iNaturalist has developed a new geo model. So it used before a system of grid cells to predict observations. So in addition to the images of the plants used in the machine learning model, it's also using geographic location and elevation to try to predict what species may occur where. And so that feeds into this species suggestion that appear on the app when you upload a photo. So these models are continuing to improve, and some of them work better than others for a certain species, but it's nice to see that continuing to develop. So with that, I'd be happy to take any questions.